Hello students, today I am continuing with the metabolism of nucleotides. I shall commence with salvage pathway of purine synthesis. Another pathway of purine biosynthesis that we call it salvage pathway. Salvage pathway, the three purine bases derived from the normal turnover of nucleic acids are recycled in tissues incapable of de novo synthesis. Tissues like that occurs in tissues like brain, RBC, and neutrophils. Okay. So they have where de novo synthesis is not operative, where these sites, in these tissues, brain, RBC, and neutrophil will get the salvage pathways, and these the free purine bases are reconverted to their corresponding nucleotides, and the process is known as salvage pathway. And salvage pathway, you see, this requires less energy compared to the de novo synthesis. You see these three bases: adenine, guanine, hypoxanthine. So these are the adenine, guanine, they are free bases. And PRPP, so PRPP plays a crucial role in the salvage pathways by acting as the donor. So PRPP is the donor of the ribose 5-phosphate. So that is important. Adenine, this enzyme you see, APRT, adenine phosphoribosyl transferase, and catalyzes the formation of AMP from adenine. So adenine phosphoribosyl transferase that catalyzes the reaction of the synthesis of AMP from adenine, and using PRPP, the donor of the ribose 5-phosphate. HGPRP, that is hypoxanthine guanine phosphoribosyl transferase, the enzyme catalyzes these two steps. HGPRT that catalyzes guanine to GMP and from hypoxanthine to IMP, so that these free bases are converted to the corresponding nucleotides. Now HGPRT catalyzes similar, convert guanine and hypoxanthine to this, and synthesis, now the synthesis of deoxyribonucleotides. Like deoxyribonucleotides, they are building blocks on DNA. They are not synthesized by the de novo, but they are formed from their corresponding ribonucleotides. Okay. So deoxyribonucleotides are not synthesized as such, but they are synthesized. First, the ribonucleotides are synthesized by de novo synthesis, and deoxyribonucleotides are synthesized from the ribonucleotides. Now, ribonucleotides, you see, undergo reduction at the C2 position, where the hydroxyl group on the ribose moiety is replaced by the hydrogen atom. So the enzyme is ribonucleotide reductase. The catalyzes reaction for all these 4 ribonucleotides. So from all these ribonucleotides, all these deoxyribonucleotides are formed, deoxyribonucleoside 5-phosphates. So reaction mechanism, the ribonucleotide reductase complex. This complex is a multi-subunit enzyme possessing allosteric sites. So ribonucleotide reductase complex is a multi-enzyme and the multi-subunit enzyme possessing allosteric sites. The sulfidyl group of the enzyme provides the hydrogen atoms that required for the reduction. A protein cofactor thyrodoxine supplies the reducing equivalents. So that reduced thyroxine that supplies the reducing equivalents. Okay, and so now thyroxine is oxidized in the process. It supplies the reducing equivalents and it is oxidized process, and it is reduced back to this reduced thyroxine with the help of this enzyme thyrodoxine reductase, and that reducing equivalence is donated by NADPH, reduced NADP, that is NADPH and H. Now again this thyrodoxine is reduced and NADPH is oxidized. So this cycle is maintained. The thyrodoxine oxidized and reduced. Okay, so there is recycling is done. So the activity of ribonucleotide reductase is regulated by allosteric modulators, which ensure that synthesis of deoxyribonucleotides occur. only when the cells are actively synthesizing DNA and preparation, that is the preparation for the cell division. So this is about the synthesis of the purine. Now come to the degradation of purine nucleotides. Now degradation of purine nucleotides of a cell undergo continual under same nucleotides of a cell undergo continual turnover. Nucleotides are hydrolyzed to nucleosides by the nucleotidases, which are further degraded to free bases and ribose or ribose phosphate by the nucleoside phosphorylases. And some of the bases are used to form nucleotides by salvage pathways, and others are degraded and to products that are excreted. The degradation of purine nucleotides finally results in the formation of, you see, this uric acid. So uric acid is the end product of purine catabolism or degradation in the humans and other primates. So the purine degradation leads to finally the uric acid. Uric acid is the end product of purine catabolism of humans and other primates. The pathway depicting the degradation of is, you see, this pathway, the AMP, IMP and GMP. So nucleotide monophosphates, the AMP, IMP and GMP, they are converted to their corresponding nucleosides, adenosine, inosine and guanosine by the nucleotidases. This is the nucleotidases. AMP and adenosine, AMP and adenosine, they are deaminated by this AMP deaminase and adenosine deaminase to IMP, inosine monophosphate and inosine. So the AMP to AMP deaminase to IMP and adenosine to adenosine deaminase to inosine. Okay. The nucleosides inosine and guanosine. So from GMP, nucleotide is from guanosine. Inosine and guanosine, they are cleaved by this purine nucleoside phosphorylase, the PNP. So from inosine to hypoxanthine, and from guanosine becomes this guanine and ribose 1 phosphate and ribose 1 phosphate. Ribose 1 phosphate is isomerized 5 phosphate, which is utilized in the synthesis of PRPP. It should be noted that adenosine is not degraded by PNP, but it is converted to inosine and further metabolized. And guanine is deaminated. So guanine is deaminated to xanthine. So it is what is above mentioned reaction. The adenine nucleotides, so from adenine nucleotides end up with hypoxanthine and guanine nucleotides are degraded to xanthine. So from adenine nucleotides comes to this hypoxanthine and guanine nucleotides comes to this xanthine. Okay. And xanthine oxidase is an important enzyme. You see, xanthine oxidase converts hypoxanthine to xanthine and from xanthine to uric acid. So that xanthine oxidase are found exclusively in the liver and small intestine. Xanthine oxidase contains electron transferring molecules such as FAD, molybdenum and iron sulfur clusters as phosphate group. Molecular oxygen, we say the molecular oxygen is used and that find an electron acceptor in the reaction and is converted to hydrogen peroxide. It is a free radical and potentially dangerous to the tissues, hence that is decomposed by catalase to water and oxygen. Now comes to uric acid. So uric acid is the end product of purine catabolism in humans. A normal adult human excretes 0.5 to 0.7 gram of uric acid daily, which is formed both from the dietary purines and from the turnover of purine nucleotides of nucleic acids. Uric acid is also excreted in product of in other primates and birds. It is further degraded in other species. Uric acid acts as a natural antioxidant in the biological system. In the process, it gets converted to allantoin. Now, uric acid, you see, is the end product of purine catabolism. Is metabolism. Purines are obtained from three sources. You will get in human diet, you get uric acid, you get purines obtained from this human diet, the degradation of endogenous, breakdown of endogenous nucleides, and the de novo synthesis. You will get the three sources of purine: diet, breakdown of endogenous purine, and de novo synthesis. This is the purine you will get. Now, breakdown will lead to uric acid. Now, uric acids and their dietary sources are about contribute to about 300 milligram and daily synthesis every 400 milligram. Total body pool of urate in men is 1200 milligram. In women, approximately 600 milligram. And you get this uric acid. Total 2/3 of this total uric acid excreted by the kidneys, 2/3, and rest by the gut, urocolysis. Urate excreted in the gut is metabolized by carbon dioxide and urocolysis carbon dioxide and ammonia by gut bacteria, so that urocolysis occurs in the gut, the carbon dioxide and ammonia. But 2/3 are excreted through this kidney. In chronic renal failure, the blood concentration of uric acid rises when the glomerular filtration falls drastically. Normal blood level of uric acid or serum level 3 to 7 milligram per deciliter in males and 2 to 6 milligram per deciliter in females. And disorders of purine, most of the disorders of purine metabolism are associated with hyperuricemia. A term refers to elevated levels of uric acid, that is in the blood, above 7 milligram per deciliter in men and 6 milligram per deciliter in women. Hyperuricemia occurs maybe due to increased production of uric acid, decreased excretion, or combination of both. There are several underlying causes of hyperuricemia: excessive alcohol consumption, inherited metabolic disorders, kidney disease, malignancy, preeclampsia. Lower uric acid level or hyperuricemia are uncommon and have little clinical significance. So, hyperuricemia is usually associated with severe liver disease or renal tubular defects. Now, what we call that hyperuricemia at body pH, normally the hyperuricemia is typically asymptomatic and does not lead to complication, but sometimes it leads to complication, or if hyperuricemia maintained, it leads to a disease called gout. But typically, in most of the cases, it is asymptomatic. So, hyperuricemia does not always lead to gout, but gout is always preceded by hyperuricemia. A body pH, uric acid occurs in the ionized form, and that is urate. In extracellular fluid, sodium is the major cation. So, uric acid occurs in combination with sodium, where sodium is major cation. Uric acid occurs in combination with sodium and its salt sodium, salt called monosodium urate. So, hyperuricemia can lead to deposition of monosodium urate crystals in the joints and an inflammatory response to these crystals causing first acute and then progresses to chronic gouty arthritis. And nodular masses, and that urate crystals may occur in the synovium, synovial fluid joints, causing excruciating pain associated with swelling and redness. The inflammatory condition popularly is called the gouty arthritis. Nodular masses of monosodium urate crystals may be deposited in the soft tissues and are known as tophi. Sometimes gout is associated with formation of also the uric acid crystal stones in the urinary tract called urolithiasis. Typically, gouty arthritis affects the first metatarsophalangeal joint, big toe, but other joints may also be affected. At 30 degrees centigrade, the solubility of uric acid is lowered. Uric acid is deposited in the cooler areas of the body to cause tophi. Gouty attacks may be precipitated by high purine diet and increased intake of alcohol. The definitive diagnosis of gout requires aspiration and examination of the synovial fluid from an affected joint. That is, you can take material from tophus also using polarized light microscopy and to confirm the presence of needle-shaped monosodium urate crystals. And gout, we can call it as a primary gout or secondary gout. The gout can be divided as, we can call it primary when either it is idiopathic, the cause is unknown, or as a result of inborn errors of metabolism. The metabolic disorders that mostly occur due to inherited enzyme defects result in increased synthesis of purine nucleotides, leading to marked hyperuricemia and gout. The biochemical defects altering uric acid homeostasis are the causes of primary gout are hyperactivity of the PRPP, phosphoribosyl pyrophosphate synthetase. PRPP is a critical regulatory intermediate in the pathway of purine nucleotide. So it is a critical regulatory intermediate in the pathway of purine nucleotide synthesis, de novo synthesis. The enzyme PRPP synthetase is normally under allosteric control. However, variant forms of PRPP synthetase they are not subjected to the allosteric regulation, causing increased production of purines, which in turn results in hyperuricemia. Another is called the superactivity of PRPP glutamyl aminotransferase. And this abnormal variant of enzyme is not sensitive to the usual. Normally, this is under feedback regulation, but abnormal variant of the enzyme is not sensitive to usual feedback regulation, and that results in the overproduction of purine nucleotides. And HGPRT deficiency, hypoxanthine guanine phosphoribosyl transferase is an enzyme of the purine. This is the enzyme of purine salvage pathway, and that gives the disease called Lesnian syndrome. And the disorder causes increased synthesis of purine nucleotides by two mechanisms. That first, it limits the utilization of purines, guanine, hypoxanthine, and PRPP by the salvage pathway that diverts PRPP for the synthesis of purine nucleotides. Second, the diminished synthesis of IMP and GMP by the salvage pathway impairs the feedback regulation of de novo synthesis of purine nucleotides and resulting in their overproduction. Another is the glucose 6 phosphatase deficiency. The condition is also called the type 1 glycogen storage disease or von Gierke's disease, in which glucose 6 phosphate cannot be converted to the glucose due to deficiency of the enzyme glucose 6 phosphatase. and this increases the availability of glucose 6 phosphate to the HMP shunt pathway, resulting in excess formation of this ribose 5 phosphate and PRPP, which in turn causes purine overproduction and hyperuricemia. And lactic acidosis is also associated with von Wieren's disease, that is associated impairs uric acid excretion, because uric acid and lactic acid that shares a common excretory pathway transport and transport system. So lactic acidosis also in that occurs in the von Wieren's disease, and that also associated with the impairs the uric acid excretion through the kidneys. That also leads to hyperuricemia. Another is abnormal variant of glutathione reductase. The enzyme glutathione reductase converts oxidized glutathione to reduced glutathione utilizing reduced NADP, that is NADPH, which is generated by the HMP shunt pathway. Abnormal activity of the enzyme causes increased production of ribose 5 phosphate, thereby PRPP eventually leads to hyperuricemia. Now, among these, the glucose 6 phosphatase deficiency. Glucose 6 phosphatase deficiency is an autosomal recessive disorder, autosomal recessive trait, and both AGPRT deficiency and hyperactivity of PRPP synthetase are X-linked recessive. They are AGPRT and PRPP X-linked recessive, and glucose 6 phosphatase is an autosomal recessive trait. Now come to the secondary gout. Secondary gout, manifestation of gouty arthritis that occurs due to secondary to certain other clinical conditions, causing, we called it, increased synthesis or decrease or reduce excretion of uric acid. You can say, now if we say increase production of uric acid, that is occurs due to the rapid increased cell growth and turnover in conditions like rapidly growing malignant tissues like leukemias, lymphoma, polycythemia. That may be due to increased tissue breakdown after treatment of large malignant tumors. That may be due to also increased tissue damage due to trauma and raised rate of catabolism as in starvation. In these, all these things can lead to increased production of uric acid. Another thing is the reduced excretion rate. Reduced excretion uric acid, renal failure, there may be, and then treatment with thiazide diuretics which inhibit the tubular secretion of uric acid, lactic acidosis and ketoacidosis, and that is interference of the tubular secretion of uric acid. Now treatment of gout. The treatment, we can treat this gout. Several approaches. Number one, reduce dietary purine intake and restrict alcohol. So reduced dietary purine intake and restrict alcohol, number one. Uricosuric drugs, increase renal excretion of urate by uricosuric drugs, which decreases the reabsorption of uric acid from the kidney tubules. Like example, you can uricosuric drug, the probenecid, the uricosuric drug that decreases the reabsorption of uric acid from renal tubules. A drug called allopurinol. That allopurinol is an analog of hypoxanthine, and it's a competitive inhibitor. Allopurinol is a competitive inhibitor of xanthine oxidase. And xanthine oxidase, converts hypoxanthine to xanthine and xanthine to uric acid. Now, xanthine and hypoxanthine, okay, they are more soluble, so they are excreted more easily. But uric acid is more insoluble, and that's why that form the complications and the deposited from the crystals and all this tophi, all this. But xanthine and hypoxanthine, they are accumulated, and because of the inhibition of xanthine oxidase, so they are more soluble, they are gets excreted, so they are less likely, or actually they are less likely to initiate inflammatory responses. And xanthinosin, in addition, it converts allopurinol to alloxanthine, which is more effective inhibitor of the xanthine oxidase. And so alloxanthine that is formed from allopurinol with the effect of this xanthine oxidase that forms is a powerful inhibitor, more powerful inhibitor, and that is an example of suicide inhibition. And others is that colchicine and the anti-inflammatory agent, and also the NSAIDs like indomethacin. You can use steroids also or prednisolone for this to control the inflammation in this gouty arthritis. So these are the modes of treatment of the gout. Another disease called Lesnian syndrome. Lesnian syndrome can say this X-linked inherited disorder, another genetic disorder, and that is characterized by this. There is a deficiency of hypoxanthine guanine phosphorybosyltransferase transferase, of salvage pathway enzyme. It was first described in 1964 by a medical student, Michael Liss. So that's why, and his professor, William Nihan. That's why it is called Liss and Liss-Nihan. And that's why, and that is characterized by spasticity, mental retardation, self-mutilation, hyperuricemia, nephrolithiasis, and gout develops later in the life. Okay, so this is about, and another is severe combined deficiency of the enzyme ADA, adenosine deaminase, will lead to is of course the defect both T and B cell. That's because in this patient DATP levels elevated inhibits the enzyme ribonucleotide reductase, and the DNA replication is adversely affected mechanism. This is treatment is only severe B cell and T cell T lymphocyte T lymphocyte production is inhibited, and the combined immunodeficiency develops, and the treatment is the gene therapy. So we have completed about the purine metabolism. Now comes to this pyrimidine metabolism, and also like purine, it has a de novo synthesis of pyrimidine nucleotides and the salvage pathway. But unlike the formation of purine bases and that occur upon a pre-existing ribose phosphate, the pyrimidine ring is assembled as free pyrimidine first and then attached to the ribose phosphate forming pyrimidine nucleotide. So in case of purine, that basis, that synthesis that occurs upon a pre-existing ribose phosphate, but pyrimidine ring is assembled as free pyrimidine first and then attached to the ribose phosphate forming pyrimidine nucleotide. The framework of pyrimidine base is assembled from bicarbonates and amino acid aspartate and glutamate. Now first the de novo synthesis of pyrimidine nucleotides. You see this the pyrimidine ring, this the contribution, this C2 and C2, that is from bicarbonate carbon dioxide, and N3 that is from glutamine. Okay, and rest of the atoms that are contributed by, that is aspartate, the amino acid aspartate. Okay, so see the contribution. So de novo synthesis. Now you see the steps of the synthesis. The first step in de novo pyrimidine biosynthesis, the formation of carbamoyl phosphate. Carbamoyl phosphate from bicarbonate, and I can see bicarbonate and ammonia that is derived from glutamine. That is glutamine is ammonia is derived from this glutamine, utilizing 2 molecules of ATP. So this is CPS2, CPS carbamoyl phosphate 2, and ATP is also get energy from this reaction. And CPS1 we have got, this CPS1 we have seen in urea cycle, the first step So the CPS1 is the mitochondrial enzyme, but CPS2 is a cytosolic enzyme. So the enzyme present in the cytosol, and CPS1 in the urea cycle that is present in the mitochondria. The carboyl phosphate, and that is combines with the amino acid aspartate to form carbamoyl aspartate. The carbamoyl aspartate, the carbamoyl aspartate. Reaction catalyzed by allosteric enzyme, that aspartate transcarbamylase. Aspartate transcarbamylase. The C2 and N3 are derived from this carbamoyl phosphate, the C2 and N3. All the rest are derived from the aspartate. Okay. So aspartate, so C2 and N3 from carboyl phosphate, and rest are from this aspartate. Pyrimidine ring closure occurs in step 3. Pyrimidine ring closure occurs in the step with the covalent bond formation between third nitrogen and fourth carbon, dihydroorotate. Okay. Dihydroorotate catalyzes the reaction and formation of dihydroorotate. Fourth step, step 4, dehydrogenase dependent on NAD. So dihydroorotate dehydrogenase dependent on NAD plus removes hydrogen atoms from C5 and C6, forming orotic acid. Is a formation of orotate or orotic acid. And leflunomide, an immunosuppressive drug which is used in the treatment of rheumatoid arthritis, inhibits pyrimidine synthesis. In this step 4, this dehydrogenase. Step 4, the leflunomide, immunosuppressive drug, is used in the treatment of rheumatoid arthritis, inhibits this pyrimidine synthesis. Step 5, the PRPP donates ribose 5-phosphate, and that is now added to orotate. Now add PRPP to produce OMP, orotidine, the formation of this orotidine monophosphate. So PRPP is added orotidine monophosphate catalyzed by orotate phosphoribosyl transferase. Step 6, that is C7 of OMP, is eliminated as carbon dioxide. So that is OMP decarboxylase. Okay. OMP decarboxylase producing UMP, so producing uridine monophosphate. A kinase enzyme phosphorylates UMP, forming kinase. That is UDP. UMP kinase forms the UMP to UDP, uridine diphosphate. Okay. Now UDP, that serves as precursors of all other pyrimidine nucleotides like UTP, CTP, CTP, DUDP, and TMP. So that is, you can say, that is the precursor UDP, that UTP, CTP, TMP, and DUMP. So all these from UDP will get DUMP, TMP, CTP, and UTP. Now, so this will get from the ribonucleotide reductase and that DUDP, and then thymidylate synthetase and TMP, and then from kinase you will get ETP, and TMP synthetase you will get CTP. Now come to the regulation of pyrimidine synthesis. The enzymes catalyzing first 3 steps, CPS2, aspartate transcarbamylase, dihydrogenase, are the 3 different domains of the same protein. Okay, the functional units. Likewise, the enzyme catalyzing is step 5 and 6 of the domains of single protein, the bifunctional enzyme. The organization of the multiple domains in a single protein synchronizes and coordinates the metabolic pathways. CPS2 catalyzing the first step is inhibited by UTP and activated by PRPP and ATP in mammalian organisms. Aspartate transcarbamylase catalyzing the step 2 is inhibited by the end product CTP, that is the feedback mechanism, and this is the rate limiting step in bacteria. OMP decarboxylase in step 6 is inhibited by UMP. Now the salvage pathway, the mechanism is similar to that of purines, where pyrimidines are converted to their respective nucleotides by pyrimidine phosphoribosyl transferase, that is PRPP, and PRPP supplies the ribose 5-phosphate. degradation of pyrimidine nucleotides. Unlike the end products of purine catabolism, they are which are slightly soluble, that is uric acid, but end products of pyrimidine catabolism are overtly water soluble. So pyrimidines are finally degraded to this carbon dioxide, ammonia, beta alanine, and beta amino isobutyrate. Beta alanine and beta amino isobutyrate, they are non-standard amino acids which undergo transamination to form succinyl CoA. Increased levels of beta amino isobutyrate are excreted in urine in leukemias and overexposure to X-rays due to destruction of DNA. It should be noted that beta amino isobutyrate is exclusively derived from thymine, which is a component of DNA. Pseudouridine, an unusual nucleotide that occurs in tRNA, is not metabolized by human enzymes and excreted in the urine unchanged. Now come to the disorders of pyrimidine metabolism. Okay, orotic aciduria. It is a very rare metabolic disorder characterized by increased orotic acid excretion. It is an autosomal recessive disease and biochemical defect in either or both enzymes like orotate phosphoribosyl transferase and OMP decarboxyl. Either or both of these enzymes defect occurs, characterized by the growth retardation, severe anemia, increased orotic acid excretion, and this can be successfully treated uh, by giving cytidine or uridine. Now you can think of some oncological relation. Anti-cancer drug, the drug called 6-azaluridine inhibits the step 6, thus blocking the pyrimidine synthesis. Methotrexate inhibits the enzyme tetrahydrofolate reductase and thereby adversely affects the formation of tetrahydrofolate, and tetrahydrofolate is required for the step 8, and 5-fluoroacyl competitively inhibits the enzyme thymidylate synthetase. All these substances are effectively used as the anti-cancer drugs, okay, because they inhibit the nucleic acid synthesis and they hamper the cell division. So I am concluding the topic of nucleic acid chemistry and metabolism.